Thank you very, very much, everyone, for joining us this afternoon to hear from our expert panel about the future evolution of laboratory working and what it's taken to get safely back to the lab and ensure research continues. I will be very brief. I'm Sarah Bruce-White from MedCity, who manage the Advanced Therapy Networks on behalf of London Advanced Therapies. If I can now hand over to Dr. Steve Walker from King's College London, who joined London Advanced Therapies to manage project programmes and institutional partnerships in this area. Steve has an academic background in biological sciences and a PhD in experimental hypertension, and over 30 years R&D experience gained through senior level positions with large pharma companies. Joining Steve is Dr. Nicoletta Charolidi. She's the manager of Imperial College Centre of Excellence in Advanced Therapeutics and one of the knowledge exchange practitioners within London Advanced Therapies. Nicoletta is a molecular vascular biologist by training, having gained her PhD in this area and having previously worked at both University College London and St George's University of London. Stephen, Nicoletta, if I can hand over to you, thank you very, very much.
Thank you, Sarah. So I'll start. My name is Steve Walker, and I would like to welcome you all to the latest London Advanced Therapies Advanced Therapy Network event, which is focused, as we've heard, on future evolution of lab work. Just a little bit of background before we get going. In 2018, key leaders from King's College London, Imperial and UCL started to put together a joint program designed to accelerate research, the whole research to commercialization pathway across cell and gene therapy activities in order to benefit patients. And with the support of a Connecting Capabilities Fund Research England grant, London Advanced Therapies was formed. The director of London Advanced Therapies is Francesca Glubich, who's joining us tonight and is in the audience. Now, London Advanced Therapies consists of a whole raft of initiatives delivered through a whole range of partners, including the London universities, as I've mentioned, King's, UCL, Imperial, but also others such as QMUL, the NHS and the NIHR, the Cell and Gene Therapy Catapult and the Advanced Therapy Treatment Centres, industry and other players. And the sort of activities that we get involved with includes knowledge exchange. We've got seed funding for a range of project initiatives across advanced therapies. And one of the, and also we're mapping the advanced therapies expertise that there is across London. So it's obviously a network event like this is really important. So one of the many initiatives is what we call the Advanced Therapies Network, or ATN, and that's delivered in partnership with MedCity. The ATN is a seminar series to enable people with expertise and knowledge across the whole spectrum of advanced therapies to share their ideas and expertise, etc. And it also creates a networking opportunity for people who are working in the field to be able to share experiences, share knowledge, and create new connections and collaborations, hopefully. So this ATN meeting, the latest one, is focused on the future evolution of lab work. And before I hand over to my colleague Nicoletta, who will take you through the aims of tonight's session, I would just like to say a thank you to our MedCity colleagues, Rick Patel, and especially to Sarah Bruce-White, who you've heard from, for their hard work putting the meeting together tonight. Sarah's done a great job actually of pulling us together, ensuring the meeting, and hopefully enabling us to deliver a remote event on Zoom, as is the way at current climate. So Nicoletta, if I can hand over to you, please.
Yes, thank you, Steve. Hello, everybody, and welcome to this event. We very much wanted to put together a meeting that concentrates on all the positive outcomes from the pressures of business as usual, even under the threat of COVID-19. And we believe that there will be a time that we're going to look back and realize that this extraordinary scenario we're living at the moment has helped us. We made us rethink, reorganize, place the environment as a priority, and actually made us take immediate action. We will have become more resilient, I want to believe, and more inventive. Because we are on our toes in terms of the economy and the environment, maybe we can increase our efficiency. And today, for this reason, we're going to look at two talks from academia, one from academia to see how academia did things in order to respond to the COVID-19 threat, and how that the industry, actually the collective of entrepreneurs did, and the health sector. And then we're going to look into some case studies which can come as a response to what needs were created and how we can keep adapting what inventions we can come with, so we can help overall efficiency of operations. Thank you very much again, and back to Steve introducing the keynote speaker.
Great, so let's get started. So it's my great pleasure to introduce Professor Mike Curtis, who is our keynote speaker for tonight. So Mike is Executive Dean for the Faculty of Dentistry, Oral and Craniofacial Sciences and Professor of Microbiology at King's College London. And one of the other things that Mike has been doing as Executive Dean for the Health Faculties at King's, he's been responsible for the return to work across the campus since our last lockdown in the spring. So Mike, over to you.
Okay, thank you very much, Steve, and thanks to the organisers of this meeting and the topic, which I think is a really important one and one we need to be having a conversation about. I'm going to present an overview presentation. It'll be quite brief. I think the meat of the meeting will actually be in the 4 talks that follow. And as Nicoletta has said, there are 2 talks from individuals who've had a real operational role in managing the working in laboratory environments during the last 9 months since the first lockdown in March. And then I think 2 really exciting talks on new developments in laboratory working, and actually ones that might get catapulted forward as a result of some of the lessons we've learned over the last 7 months. My role is simply to give a few thoughts at the beginning from the perspective of somebody who leads an academic institution in the health faculties at King's, and also some lessons learned from trying to bring 30,000 students and 10,000 staff back into 60 buildings at King's over the course of the summer. So what I'll start with, can I have the next slide, please? Is the areas I'm going to cover are 3. So what are the drivers for change in this area of laboratory working? What my view of some of the key lessons we've learned over the last few months? And then finally, and these are very much top of the head thoughts on the implications of some of this to how we are going to operate in the future. So next slide, please. So in terms of drivers for change, I don't know if any of you have been following Jonathan Friedland podcasts that are now available on BBC iPlayer, which are just 15-minute vignettes on the so-called way forward, which summarise what happens after disasters have struck society. And there's a really interesting one on the Second World War, but there's a really interesting one, I think, from the perspective of this meeting this evening, on what were the consequences of the pandemic flu in 1918. How did that change the way things worked? And as Jonathan Friedland points out, there are 2 ways in which you respond to a crisis. First one is that everybody just runs around, panics, eventually gets things sorted out, everything goes back to normal, everybody becomes complacent, and then it happens again. And the same, then you go round and round that circle. And the other outcome is that actually people think about what's happened and change the way they operate on a routine basis. And the examples from 1918 flu, I think, are really interesting because, you know, at that time in 1918, viruses were barely understood. Everybody thought it was a bacterial infection. We didn't have properly established public health systems. That changed. The epidemiology as a discipline really came into its own, understanding the spread of disease. And obviously, it was the beginning of the virology heyday. An interesting consequence of it, actually, was that when the antibiotics were discovered, they were discovered because they were looking for the causative agent of flu. thinking it was a bacterium, and it was an incidental finding that the penicillin grew on that plate. So there are many and varied consequences of human disasters. I hope that what's happened over the last 6, 7, 9 months will also lead to some significant changes. And the reason I put that second point in there, education, is that I think that has already been influenced massively by the last 7 months. The idea that we'll go back to huge lecture theatres with 400 students, the idea that we will be doing everything face to face, when the experience tells us that actually learning online is both better for students, it's certainly more challenging and more presents innovative challenges for our staff, but ultimately the educational experience can be improved. And the people involved in education are absolutely now committed to learning the lessons from the last 9 months, because they've had to adapt in order to deliver their education. As far as I can tell, the conversation around research has been much less active, and which is why I think, you know, this evening's presentations are really important, because this is a key moment, I think, in thinking about how we work. The third point on there is a culture of work. And there's no question that how the workplace operates has dramatically changed, and I think will continue to change as a result of this. You know, I come to work from North London, and I pass through the city when I arrive at London Bridge every day. And the difference between now and 9 months ago is just astonishing. I think all of the banks, the insurance companies, and all the other offices have decided it's a far easier prospect to work from home. Many of them, several of them at least, are thinking about their future and whether they need all that office space in a very expensive part of London. Our experience in the academic arena is that people actually are very, very productive, or can be very, very productive from a work environment, but it needs to be from a home environment, but it needs to be balanced with access to colleagues and the ability to meet colleagues face-to-face where possible. And then the last driver is around efficiency. And you'll hear more from some of the later speakers about the efficiency gains that have taken place over the last period, particularly around the use of shared facilities, making better use of core facilities, booking systems for equipment, and so on and so forth. Having said all that, I think much of what I'm going to speak about refers to the academic workplace. I think in the industrial sector, in pharmaceutical companies and so on and so forth, and startup biotech, they've already adapted before any of this happened. They've already learned the lessons of efficiency and of many of the things that you're going to hear about today. So I think this will, for my mind, usher in a bit of a catch-up period for the academic environment, and we'll learn lessons from one another as we do that. Next slide, please. I can't give this talk without talking about business continuity planning, because that has actually been the saviour of most organisations, that they've had plans that they can go to in the event of a disaster. And this is something that, you know, in day-to-day life working in a lab, business continuity is not something at the front of everybody's mind. You occasionally see a spreadsheet with a few kind of red marks on it that you need to do something about. But for the most part, it's something that operates very much in the background. But business continuity planning, health and safety, HR, and all of those other areas that kind of operate in the back are absolutely fundamental to making sure an institution operates. And it came as a kind of revelation to me, I suppose, that working with teams involved in BCP, health and safety and so on, have been absolutely critical to, in order to get our university back up and running on the campus. You'll hear quite a bit about the advances we've made in digital working, and that's not only through Zoom calls like this one and Teams and so on. It's just the whole concept of working digitally. Very few academic laboratories at the moment operate using electronic notebooks, electronic lab books. but the ability to use that form of data storage to be able to share information with your PI who's based 100 miles away or wherever on a live basis is actually absolutely essential, I feel, if we're going to make the most of having a limited number of staff in at any one time and their PI based elsewhere. So I'm almost certain that anybody involved in electronic notebooks is going to be very well placed, let's say, in a business sense in the coming months. Space and facilities is another one that we've learnt in the academic sector, and I think we're catching up now with the biotech sector, that the way we use space and the way we use our facilities can be much more efficient. The simple example is the use of core facilities, but it extends to every piece of major equipment in your laboratory area. You can keep that equipment busy for a long, long time if you have bucket systems that allow people access from different laboratory areas to come and use that. So I think some of the talks you'll hear later emphasize the value of that kind of proper sharing of equipment. And it goes without saying that all of this comes with efficiency gains. One of the things that is also a key lesson, and from the downside, I suppose, is that these things don't happen automatically. And one has to invest in training of staff to get the most out of, for example, digital working and the use of shared facilities. So training is a really significant aspect. Next slide. And then what are the implications of this? And these are just thoughts from the top of my head, as I said earlier. But I think anybody building a new laboratory building now would pause at this point and think, well, what have we learnt in terms of layouts of laboratories, layouts of office space, adjacencies to areas where people can sit and talk and so on. And in many ways, this lesson has been learnt already, but not yet been fully adapted. Those of you who know the Crick up at King's Cross will already recognise that many of the kind of design features in that building really emphasise the need for having spaces where staff can sit in a reasonably comfortable environment and exchange ideas and so on. I think that's just one small aspect of it. The other area is governance. And I think there's no doubt, to my mind, the introduction of digital technology in the way that we share data, particularly around laboratory primary data, is far better served in a digital environment. And that brings with it a whole host of benefits in terms of governance of the research and accuracy of data and the ability to check data and so on. So I think our governance arrangements will also respond to this. The last one, the penultimate one, is research training. And call me old-fashioned and all of that, but my first car was a minivan, and I could disassemble every part of that minivan and put it back together again. I knew when there was a problem with it. I knew, when the power went, what the likely problem was, why it didn't steer properly, and so on and so forth. I look at my car now, and I can't do anything on it apart from start it and stop it, because it has become a black box that just goes back to the supplier. Now, I'd use that analogy because the more and more we rely on centralized facilities, on commercially supplied gels and columns and so on and so forth, the further away we get from the practice of laboratory research for those involved in it. It becomes more, shopping for the right thing and knowing which thing you need to buy, but essentially you're not troubleshooting. I thought about this, I've talked to my students about this, and they said, oh, no, that's not right, because where the art is now is actually managing data and being sophisticated in the use of computer programming, understanding the best ways to handle mega data sets. That's where the artistry is now. It's not about how well you can pour an 8 molar urea gel. It's about you really having the kind of skills and intuition and so on when it comes to big data analysis. But I still think there's something in there that delivering everything via very large single facilities takes away some of that learning about the practicalities of doing research. And then I think my last area before I stop, I'll probably run over time, I'm sorry, is around cooperation and collaboration. And I perceive that as being one of the really major steps forward that we've managed over this last period. In our environment, people are far more cooperative about use of facilities because they have to be. And people and groups are far more collaborative. And just having the ability to beam in a world expert into your lab group on a regular basis without having to pay an enormous air bill is now commonplace, you know, and I really struggle to see that we'll go back away from that. The last area, and I haven't put it up there, is another important agenda item, certainly in the academic sector, and I think growing in society, is the whole equality, diversity and inclusion area. And many of the things I've talked about lend themselves very well to that area. The homeworking goes without saying, offers real benefits to those with caring responsibilities. The way we design our estate, the way we enable access to the estate obviously brings benefits to those with disabilities and so on. So I think, you know, the range of things that we could talk about are very, very extensive. And I'm just happy to be part of the conversation to start with. And with that, I will hand back over to Steve. Are you picking up the baton now?
Nicoletta, yes.
Yeah,
Thank you very much.
thank you.
Thank you very much, Mike. So it's my pleasure to introduce the next speaker. Is Alison Hunter. Alison Hunter is the Technical Operations Manager at the Department of Life Sciences at Imperial College. She is an expert in managing complex multi-site operations with experience in sustainability, having been very active with the Green Lab initiative and having been recognized with achievement awards from King's College London and S-Lab. Alison holds an MSc in biomolecular organization and is a government stakeholder and active contributor in a BEAS initiative for scoping laboratory cold storage standards. And so, Alison, over to you.
Thanks for that lovely introduction, Nicoletta. Yes, I'm Alison Hunter, and here are some of our labs in Sir Alexander Fleming building on the South Kensington campus that I look after. Next slide, please, Sarah. And as Nicoletta said, I've always worked in University of London labs at about 3 different institutions. So I was a contract researcher before I went into lab management. I look after staff development and sustainability. Next slide, please. So I'm going to describe to you how we kept going in our labs. So a Department of Life Sciences, and we would be the Department of Dead Sciences very quickly if we couldn't come in and keep all of our stocks alive. So we'll talk about the initial lockdown process and then how we gradually reopened our labs for our PhD students and grant-funded research projects. Some of the benefits that we found. I'll talk a little bit more about lab sustainability issues and give you some resources at the end on those. Next slide, please. So once lockdown was announced, the first thing I did was put out some really swift communications to reassure everybody in the department, because I anticipated quite a lot of panic. And so that was about what's happening with the facilities, the services and the technical staffing during the lockdown and what they could expect. So the business critical processes for them is, can I get cylinders for my CO2 incubators? Can I get liquid nitrogen? Can I get media autoclaved? What are you doing with my biohazardous waste? Is stores open? So by anticipating all of those questions and just giving a reassuring email about what would happen for all of those, just stemmed the email tsunami of panic, and that seemed to work very well. And we had remote Teams meetings to mop up any questions that anybody had. I've got 500 staff in the department, 200 postgraduate students, and about 800 undergraduate and master's students. And we occupy 16,000 square metres of laboratory space across 2 campuses, and we have 70 controlled environment rooms. So there was a lot of demand for people to come in. You'll see our mosquito insectary on the top picture. We've got mesocosms, which are ponds. We're looking at different species growth as they inhabit those ponds. And the last lab is a protein structure lab. And we have a huge variety of other labs as well. So everybody was quite keen to have their projects and have access to come in and keep everything going. So we had an access request form, and we did an academic and technical triage. Who could come in, what could they do, which projects were we going to prioritise? And I think we got that down to about 200 requests. Some people just had to come in to shut down within 2 days, 4 days, 2 weeks, or ongoing. And then we reviewed that process after about 3 weeks for any other requests. So staff had to be flexible in terms of when they could come in, obviously avoiding peak hours to come in. We had special letters so that we could travel in with approval from the provost of the college. So we had multiple backups straight away. So when we requested the number of people to come in that we did, the college said that's too many. But we explained we'd already anticipated bubbles, and we had double or triple cohorts, because if somebody fell ill, the next day somebody has to come in, and we don't have time to get permissions for them to come into the lab. So we already had that in place. So having that business continuity planning at the beginning meant we could really quickly act. Working safely was really important. So we had faculty safety, college safety, and government advice to follow. And that changed throughout the lockdown as well. So keeping up on top of that to make sure everything was being done properly. And with less people on site, we maybe had 30 or 40 people in the labs initially. The potential for lone working. We used a safe zone app on our telephones so that you could immediately alert security if you're having any particular problems in an area, so that people felt a little bit more safe in the labs. On top of all of that work, several labs repurposed their research. So from a mosquito project, from cancer cells, and from laboratory environmental monitoring of water quality, they all repurposed projects to COVID-19, received grant funding for that, and we had lab refurbishments and procurement to do on top of all of those other things. Next slide, please. So by about June, we hadn't had any infections on site, so we decided that we would open up to more 10% occupancy, and we did that gradually. So we opened floor by floor at 10%, and we still didn't have any COVID spikes or infections. So that all of the measures that we put in place did work, but obviously we didn't know if it would at the beginning. We did extensive building route signage, lots of disinfection points in the kitchens and things like that. So it took quite a lot of time to find out what we were going to do with that. The college had a reopening checklist, and we worked through that. So things like flushing all the taps for Legionella. We had already regularly cleaned our ice machines and water systems, and had our statutory tests done for microbiological safety cabinets and things like that to make it easy for everyone to come back. Risk assessments and SOPs had to be done for the building and for the labs. And we increased the tech support from 2 days a week to 5 days and continued with the bubble system. We worked with the faculty safety team and produced an online safety video, which was 25 minutes. And people could sign up to come back into the labs. They had to show that they'd completed this video before they were put onto a PPMS booking system. So our departmental administrator looks after all core facilities in the department, and we have this Stratacore PPMS system for equipment booking and sharing. And she repurposed that very carefully for the labs. So you'll see in the photo the red and white stripes. That demarcates an area where you can work most of the time. And she anticipated the return in September, October, that if the COVID was still happening, how were we going to teach masters and undergraduate students? So you'll see in the bottom photograph, there's another bay in the lab. So everything was staggered 2 meters apart. And we spent quite some time measuring all of those different benches across all of our labs to make sure people were safe. For teaching purposes, you're going to have to be less than 2 meters. So we have additional face visors and disposable masks if you're at less than 2 meters. And this system is booked very well. So these spaces can be booked maybe twice a day or 5 times a day. We altered the time slots to suit the different labs. Say the Drosophila labs just needed 1 hour, other labs needed just 2 hours. And so you can get 4 or 5 different people in during the day, but they're just not all in at the same time. And in October, we had 4,976 sessions. And we reviewed and refined that all the way through. And then for the teams, I had 25 technicians. Some were furloughed, some were working from home, and some were on site or sporadically on and off site. So having team meetings with sort of not necessarily just about work, but talking about other things, funny videos, other nice walks in the countryside that you can do, virtual tours of things, so that we could all do things together and keep that team spirit going and let people who were maybe working at home or furloughed feel connected to what was going on. And we tried to rotate the staff as much as possible so that some of them at least had a chance to come to site and learn what all of the new procedures were, so that everybody was resilient and understood what the new processes were. And next slide, please. So I think everyone found the benefits of increased digital literacy and continuing professional development. For us, getting my team all onto a Teams process and putting all of the information, our building routes and waste pickup points there for everybody to see, so that if the whole tech team went down, anybody else in the labs could pick those things up and just follow everything to make sure that we didn't lose any research. Certainly for our malaria labs, it's 30 years and 150 mosquito lines. So to lose that would be really catastrophic for the department. The Science Council did a survey for technical staff in June and July and found that there was much greater profile and recognition for technical staff within universities. I think people actually realised what technicians do behind the scenes and keeping everything running. That became really obvious. And the National Technician Development Centre said technical staff themselves felt very optimistic. And the NTDC also ran Tech Meet for about 50 different universities across the country. So we could talk about all of the processes as things changed. Well, how are you doing that? What approaches are you taking? So there was a lot of collaboration and sharing of what people were trying to do to make their labs work. And I think within the lab, because of the very restricted time slots people could have, life sciences work usually is an ongoing process and things don't grow necessarily at the time you want them to. And people were helping colleagues out. So the next day they would take care of things for somebody who then couldn't book to come in the next day. A lot of sharing within that. And between the health and safety teams and the admin and estates, everybody was working together to make this work. Next slide, please. And there's increasing collaboration between university and business. And this was certainly the case for the COVID with vaccines and ventilators. Certainly our aeronautics labs started making parts for the visors for PPE because there were problems with supply chain, was completely disrupted initially. So some parts, especially for ventilators, couldn't be imported. And so they started making them themselves. Next slide, please. And so sustainability, certainly over the last 10 years, has been, becoming increasingly common to discuss. But I think in the light of the COVID, it's made us think actually about climate change and what impact do we have as scientists on the planet. Certainly, we are signing up to a lab cup system for chemical inventory. We have a huge floor footprint in our labs. And we have maybe 200 or 300 people coming to our labs every year. Everybody's ordering and buying things. So having a central inventory system is really helpful. This is the chemical waste from just one laboratory that occupies 2 bays. 80% of the chemicals that they had were no longer required or redundant or out of date. So having a system and process in place means more effective sharing. If you've got a kilogram of stuff, you can let other people from other labs just take a couple of grams. You don't need to buy another kilogram pot yourself. I think equipment sharing, Mike mentioned that earlier. We have lots of core facilities in our department, but I just put a link up to the UKRI info portal for facilities across the whole of the UK. And I think grant funded research people tend to buy equipment just for their lab for that project. And I do think we need to rethink that model for equipment sharing. And waste, there's a whole nother talk that could be had on waste, but I just put one link in there. We use this quite a lot, Warp It, to repurpose things from our lab so that somebody else gets reuse out of something that we don't want to use. And we certainly really recycle our plastics and metal and wood, all kinds of things like that. I think reducing all of the things that we send to landfill is really important. And we need to be prudent with our resources. Future funding after COVID is going to be difficult given the amount of money the government is subsidising with all the furlough scheme. And we need to ensure that we're being really prudent with the resources that we've got. Next slide, please. So just one item of equipment is freezers, because everybody's got them everywhere. And I think we need to be more active looking at all the different types of equipment that we have in our labs and asking for life cycle costing. Things that tend to be quite cheap tend to have very high running costs. So you need to look at what your electricity or water bill is going to be at the end of that. And there can be large differences across the market. Certainly for freezers, there was a huge difference for minus 80s in terms of performance. So you've got to be careful about what you're buying. And just making small changes by increasing the temperature of your freezer, you can make huge savings, and certainly carbon savings are going to increasingly be important. Freezer costs for a whole university can be at about $500,000, and I would estimate that's the same at Imperial as well. This is a HEFCE Catalyst funded study that I carried out there about 5 or 6 years ago. And what we measured when we measured the freezers, that an energy inefficient freezer costs about £1,300 per freezer per annum, and an energy efficient one can be about £270 to £350. So it makes a really big difference if you buy an energy efficient one and then manage it effectively. Next slide, please. So here just some resources that I thought might be of use. And this paper at the end, the link with the FEBS letters with the DOI.org, the connections between laboratory research and climate change, what scientists and policymakers can do to reduce environmental impacts. In your lab, you can make a difference by having a green program and look at your recycling, look at what you're ordering and how you're using it, sign up to lab efficiency. It's incredible what you can save with not really too much effort. And certainly I'm pushing for equipment standards and legislation for freezers. I don't see why I can go into John Lewis and buy a A-rated freezer, but I can't do that for a laboratory freezer because there are no standards in place. So I've been lobbying government and European Union to have more standards in place, given the high number of pieces of equipment we have out there. Work with your estates departments, make sure that the facilities that you're newly building, go to lessons learned from what people have done before, go and visit other facilities and find out what worked and what didn't, and look at the energy of the whole building. That makes a massive impact on what you do in the lab if you get the building right. And again, having institutional policies, and it's really encouraging to see UKRI have recently published a sustainability strategy as well. Climate change is going to be the next crisis we're facing, and I think it's important as scientists that we get involved and reduce our own environmental impacts. Thanks for listening, and I'd just like to introduce Dave Mudd, who's your next speaker.
Thank you very much, Alison. Thank you very much. This has been really helpful. I guess a very important consideration is to make your students also get rid of all the samples from all the freezers.
Yeah. Yeah.
We're going to move to Dave, as I said. Dave is the Director of Facilities and Estates at the Entrepreneurial Hub, the Stevenage Bioscience Catalyst. Dave has been an expert in health and safety, facilities, hard and soft, bespoke lab design and fit-out project management. David, I guess, is the man who needs to be addressed when changes are required at the workplace. So please, David, tell us what you did in Stevenage.
Okay, thank you. Thank you, Nicoletta. Thank you, Alison. SBC, for those who are not aware, SBC is located on the GSK campus at Stevenage. Currently, we have 4 buildings with around 40 companies, and they all range from virtual to startups to larger companies of around about 100 people. What I'd like to talk about today are the steps taken to keep the SBC buildings and the facilities open and operational during the lockdown, well, the first lockdown, and obviously now, as we now go into the second period of lockdown, and what preparations that we've put in place in readiness for that. at the very beginning, so going back to March this year, our companies came to us very quickly, and they realised that they needed to continue operating and requested the support to keep the facilities open. Now, working very closely with, as we're a GSK campus, GSK being our main site stakeholders, we spoke to those quickly to look at how they can provide site access for critical R&D companies and certainly manufacturing as well. So as one of the buildings we have on site is the cell and gene therapy catapult, which manufactures therapies that are taken direct to patients, and potentially are saving patients' lives out there. So it's very critical for us to ensure that we are engaged very quickly to have a seamless opening or a non-closure of our site. So from that, SPC remained open for essential business during the very first lockdown. By this ensured that the manufacturing centre and the critical R&D that was ongoing throughout the facility could still be delivered and continue. On this slide now, what we're going to go through is the processes that we put in place from the very beginning. So as Mike Curtis mentioned, one of the key lessons was about business continuity planning. We very quickly, and we put together a very comprehensive business continuity plan prior to the government lockdown, and it was in line with the government and the GSK policies and any site guidance that they were providing. So this was critical for us to ensure that GSK would allow us to keep the site open. From that, each occupier were required to then produce and provide their own management plans, stating the required essential services and what priority site accesses they required. The SPC companies responded very quickly to ensure that those required access to site were not only unduly delayed access to site, but they can continue doing the critical works that they were continuing on site. From that, we also established that we needed to ensure that any essential services were continued. So we prior to the lockdown, we were in conversations with all of our essential services. So there was a whole range of those services that we had on site. So lab gases, clinical waste, general waste, specific specialist engineer support. All of this was planned and agreed prior to the lockdown to ensure continuity for all of the people who were continuing to work on site. All of this information was then put together in our BCP, which was then shared with each of the individual occupiers within our facilities. And the request was for each of those individual organisations to provide their own business continuity plans. Now, as you probably understand, that this was probably a shock to quite a few of them. Most of them probably didn't have these in place, but we were there to be able to provide them with the support for those guys to put their plans together so that they could have continued access to site and continue to do the works that they were carrying out. Some of the other support that we put in place were that the operations team, so the SPC operations team, albeit a very small team, are supported by numerous third-party contractors, from an M&E contractor to ensure that the buildings are running, to ensure that all the plant, all of the fume extract, all of the lab waste water, the air supplies, everything is running as well as the cleaning contractors, just to ensure that as the government guidance was coming through in regards to the measures and guidelines we had to put in place, so that being social distancing, cleaning, provision of hand sanitizers, ensuring that there wasn't too many people in each of the buildings to ensure that you could continue keeping your social distancing. My team, albeit being very small, we were able to ensure that we continued on a permanent basis, providing all of the support that you'd expect for a facilities and operations team. They were always on hand should there be any queries coming through from any of the occupiers who were on site, because again, everything was new to the people on site, and they just wanted assurances that what they were doing was correct and that they weren't causing any due risk to anybody else's safety throughout the facilities. As part of that, it was very quick that there were a number of people. We had to reduce the numbers of people on site, and through that we established remote working for everybody, for our team that was on site. The same was to be said for the other organisations. We ensured that the IT provision that they had in place, that they could then link in via their VPN to keep connected to all of the critical data that was coming through from the guys who were working in the still continue to work in the labs. It was all part of their support. For our team, the office-based staff, they were able to work from home, which we prepared before the lockdown, preparing their laptops, making sure that they've got their VPN connections. As part of that, we also, as Alison mentioned as well, we had to make sure of the well-being of the staff that were working remotely, and that simple things like ensuring that we had weekly meetings in place. We, in fact, at the very early stages, we had twice weekly meetings to ensure, just engaging in topics not all work related, and just fun things, sometimes including pictures on the backs of walls, etc. Did anybody notice any changes, or somebody wearing loud and luminous t-shirts, just to keep the fun aspect going. As we then moved on, and the government guidance then started to reduce some of the restrictions that they had in place, and they started to encourage more people to come back into work, we then had to put together some additional planning documentation. So this included the generation of a return to work policy. If you'd like to do the next slide, please, Sarah. So we developed the document, which was a return to work policy, to ensure the reopening of the site for more occupiers to come on to the site. This ensured that they were looking at and addressing any of the issues that potentially this could produce with an increased number of people coming onto the SBC facilities. Going back to the early days, we only had between 30 and 40 people in 3 of the buildings, and there was a further 30-40 people working in the manufacturing centre as well. This then started to gradually increase over May and June, where we were achieving and reaching numbers of between 120 to 200 people on site. So in total, we had for the 4 buildings and on the SB facilities, we have around about 900 to 1,000 people. So we've been working to no more than a third of those at this current time, just to ensure that we can continue keeping to the social distancing within each of our buildings. This then meant that we had to then design and start implementing the site access rules to minimise, the viral transmission and the safety of our people within those within our buildings. Things that we put in place were one-way entry and exit systems, having descending and ascending stairwells depending on which building you're in and whether we were able to do that with a number of stairwells. Restricted access in lifts, for example, so not having any more than 2 people within a lift. Service corridors, making sure that people weren't stopping in the service corridors for impromptu chats or meetings, as the service corridors weren't wide enough. On top of that, we then had to start introducing more signage around the site, and these things would then re-emphasize the social distancing message to ensure that people were maintaining their 2 meters distance throughout. The message of hand washing, the wearing of masks, and simple things like reducing the amount of furniture in communal areas to ensure that people were continuing with their social distancing measures. And also we then had to push that forward through for within their actual demises as well, be it a lab and an office. So I think Alison mentioned in hers as well, regards to the taping and the appropriate signage to ensure that people were adhering to the social distancing measures, wearing a mask, be it shields, etc. So from that, we'd also look at the provision of disinfection materials throughout all the communal areas, be reception, cafe areas, etc. Weekly site updates. We ensured that the communications, any government or site stakeholder changes, they were given out on a weekly basis or as regular as any of the changes were coming through. We also had the continued liaison with the GSK management for any site regulations in line with any government guidelines. Next slide, please. Just on that there, I'm just showing some photos there of the type of measures that I'm sure most of you have seen now in regards to what's been put in place in any buildings, anywhere, ranging from reception areas with screens put up there, signage putting up for 2 meter distancing, ensuring that you've got one-way entrance and exit, booths out in the breakout areas which show examples of reductions in seating, making sure that everybody's adhering to the 2 meter rule.
Excellent. Thank you very much, Dave. Thank you. As we can see from the 2 sides of things, academia and both industry in a way had to act pretty quickly, rearrange everything. Maybe the work needed to be put in place had triple in terms of logistics and organization. So are there any applications? Are there anything that we can do to keep the momentum of our hard work keep going? Can we do anything to help everybody remain efficient? First, we're going to hear from Dr. Jean-Marie Delalande, or Jimmy, as I know him. Jimmy is a developmental biologist with a PhD from the University of France in France. Following his PhD, he came to London as a postdoc in the Royal Free Hospital and then at the Institute of Child Health. And then he also had a short stint at Hudson Cancer Institute in Salt Lake City in the States. He has followed an academic career. Currently, he's a senior lecturer in the lead of year 1 and 2 in the medical degree in Malta at Queen University, with Queen University of London. But also, Jimmy has entrepreneurial flair, and having worked for years with experimental animals, he has come up with a great idea. Let's hear more from him.
Yes, thank you, Nicoletta, and thank you for inviting me. It's very nice to be with you tonight. I'm gonna share my screen with you, and I'm going to give you a very short presentation. I hope you can see my screen now. Is that okay? Yeah. Okay. So what I want to talk to you about is following up on this idea of collaboration, and I'm going to talk to you about a website and an app that we developed and we're still working on to facilitate sharing of animals, animal tissue for research. So my two collaborators are Dr. Alan Burns, who was my boss at UCL and is now with Takeda Pharmaceutical, and Enrique, who is the developer, basically, who put the software together. What I'm going to show you. Okay. All right. So let me just start with a very quick little proposition. Imagine you're going to a country and you're visiting, and they have different slaughterhouse, and one is a chicken liver slaughterhouse, and the other one is a T-bone steak slaughterhouse. So they have either chicken or cows, and then they just extract either the liver or the delicious T-bone steak. But the kick-in is everything else goes to the bin because they're not used, because they're very specific slaughterhouse. And you would think, you know, these people are crazy. What are they doing? This is an incredible waste. But the reality of things is that research is not too far behind this kind of very wasteful situation. Recently, I was looking at this paper. It's in October, so it's last month, saying that millions of animals may be missing from scientific studies, and most animals that are used for research are not accounted for in a published paper. And it looks like animal research is extremely wasteful. If people don't like to publish negative results, they develop animal models, and if they don't work, then nothing happens. And so the idea is to bounce on collaboration. And so I worked for many years at UCL, and this is where I met Nicoletta. She was my colleague. At UCL, there's about 125,000 mice and about 1,000 different strains of them, holded by 160 different people. And when we looked at it, when we were starting to develop this app to share animal tissue, we could see that among these 1,000 mouse lines, there's about 150 that were at least duplicated, and maybe some of them were present in, there was 5 different colonies just within UCL. And this duplication just in-house is incredibly wasteful. It's both ethically and financially crazy. And we're all looking at very lean times in research right now, and we know that wasting money and not collaborating, you know, it cannot last too long. So it's possible. We have to consider that maybe some people, they maintain a colony of mice and they will harvest one tiny tissue from it and then throw everything else in the bin. And when Nicoletta was working with me at Institute of Child Health, our group was working on the pituitary, which is a tiny little gland in the brain. And I know because I collaborated with them, we would take that little piece and then everything else would go to the bin. And currently there's not many system in place, despite the fact that research institution and anyone who works on animal research is obliged under UK and European legislation to implement the 3 R's, which is replacement, reduction, and refinement of animal use. So basically, we said, well, there must be an app for that, and there wasn't an app for that. So we started creating it. So this was called OptiShare. So the first thing it does, obviously, is to share tissue. So there's a tab to share a tissue. You can enter the strain of your mice, and then you select which organ you take off, which free in the system everything else. There might be ages. If I'm a developmental biologist, so the stage of development matters a lot. And then importantly, you're going to say at which stage they're going to be available and post your resource. You can request tissue if you're interested and you're looking for a specific animal. Again, you can enter your gene of interest or whatever you're after. The organ of interest, if it's not the same as the person who posted, shared the tissue, then that will create a match. And then you can make a request at a specific time before the end of your project or leave it as open-ended. There's also a search engine that you can search by a specific strain of mice. It will tell you if someone has it. It will tell you if the organ you want is still available, and you can communicate with that person. So you can search for available resources, and that could be in-house, but that also could be in a different location in London. London is unique because we have so many different research labs there. And it's not that far to go from UCL to the Blizzard Institute where I work. So the map actually the app used Google Map to connect and tell you where exactly the institution is located. And again, I think London has so many research institutions that if we could find a match or the places, then that's very powerful. Again, so I'm cutting it very short. Normally you would get a notification if you have a match with someone. And then this app is all about facilitating collaboration and sharing resources, basically. So what we have, just to conclude, is that we have a fully operational platform that if people engage with, will create this match and collaboration. It's very resource owner centric because that with all the discussion we have, we realize that in research, people are quite protective and we need to give them the ownership of choosing to choose or not. My analogy to the slaughterhouse is not completely legit because research works very differently from an economically driven slaughterhouse. But what we need right now, and we are working still with UCL, but because of the COVID pandemic, it has taken a big step back. But the limitations we've already uncovered, that it needs to be a bit sleeker. I'll show you screen grab of the app. It's not super sleek. We want to make a phone app because for the ease of use, maybe you can be in the lab preparing for your experiment and you want to share. And lastly, it needs we need maybe people to come and join us, and obviously financial help to develop the system further, because so far it's been developed with Enrique, who's worked for free, myself and my former boss. So on this, I will stop sharing, and thank you for your attention. And this is it. I'll just. Excellent.
Thank you very much. Thank you very much, Jimmy. That looks great, and I hope it takes off a bit more. I'm considering the time, so we'll move with the second case study that comes from Moise Ibrahimji. Moise has 14 years experience working within the life science sector. He holds a degree in medical biochemistry and working within Synthase commercial healthcare. He specializes in workflows that can make a lot easier can make the life of scientists working in gene and cell therapy a lot more easier. Let's hear from Moise. Thank you.
Hi, thanks, Nicoletta. Hi, everyone. So moving swiftly on, the title of my talk today is Intelligent Automation for the Future Evolution of Laboratory Work. The current times has propelled us to make significant changes to the way we operate, and also to give thought about how our labs can be more connected to the outside world, so we can have a window into what's happening without physically going inside, but also more automated, so we spend less time processing, and tasks such as process experimentation, sample processing, which are often quite long and complicated, can be done for us in a more straightforward fashion, quickly and simply. Synthase has been actively working on this for quite some time now, And today I'll explain a little bit more about our work and its use in real life practice. So we're a scientific company founded in London, and the software we've developed is called Anther. We were originally a CRO doing huge experimental campaigns that we performed by hand. And when undergoing the natural transition to use automation, we discovered the barrier being very high and specialized. So we created a powerful piece of software originally for ourselves to facilitate our own work. However, the demand for the software became so strong, and clients wanting to know how we're being able to produce quite sophisticated experimentation and data very quickly meant that the demand for the software became so strong that we decided to pivot and make the software widely available and to enable the wider industry. We've of course kept our scientific focus and also our high throughput development labs. So this is quite unique for a software company, and hence the phrase here, designed by biologists for biologists. And just above that, you'll see that Anther is a cloud-based platform for device-agnostic experimental execution and data integration. So what does that mean exactly? Anther was created to bridge a language gap that exists. We speak in measurements, concentrations, and steps such as dilute, serial dilution, and other descriptive experimental steps. And laboratory automation speaks in code, i.e. script. So when wanting to use automation, which is a critical part of a modern and future lab, we must program. And this is quite inflexible and ridden with trial and error at best, having to really delve down into the true low-level details of your experiment and plan out each and every movement you need to do and what plasticware you need. So Anther does for you is it first translates your experiment into an already digital representation of your experiment. So you put in your scientific protocol, it translates that for you digitally, and it's against your specific hardware and the configuration that you have. And once you're happy with that, you can, it will generate the script and code for you, and you can automatically then go into the lab, press execute, or load up, prepare the reagents and press execute, which then goes ahead. And Anther knows what a qPCR is, what an ELISA is, what design of experiments is, that we've kind of worked on as optimized protocols. Following your experiment, which generally consists of some liquid handling and then analytical devices to give a readout, what Anther will do is connect to these different devices, and then what you have to do is take the results from different locations, different formats, and structure them together. And that's also a key part of working efficiently and what Anther does. With COVID in mind, the fact that it's cloud-based, you can do, of course, your translation and experimental planning remotely, as well as all the data aggregation and analysis can be done remotely as well. So let's learn about this use in the real world. So this is a case study we performed a while back with Oxford Biomedica, and this is a snapshot of a viral vector process that they wanted to optimize. You can see at the top some plasmids that encode for core structural and packaging proteins for formation of the vector particles. And the vectors here are then produced by the packaging cell line and then harvested and used to transduce a target cell line for the delivery of the genetic payload to the target cells. This is the use case that we wanted to optimize for the production of functional vector particles carrying the desired genetic payload. And to do this, they used Anther to digitally plan and execute their scripts. So just to ensure we're all aware, so what is design of experiments? And it's a powerful statistical approach in order to study how multiple factors of a process influence the overall outcome and each other. It's a robust process to process optimization, and also aims to minimize time and cost by simultaneously varying process conditions. At the top here, you see a typical DOE process of several stages, with the capabilities enabled by an ANTHA-driven optimization, but also the fact that Oxford Biomedica had a good understanding of their process. We used a two-stage DOE, a factor screen to see what factors are actually having an effect, followed by a refine and optimization stage. So what you do as a scientist is, once you've planned your experiment inside a software such as JUMP, you would export the CSV file into the front end of ANTHA and a protocol. You'd then press simulate, and ANTHA generates an in silico representation of your experiment. So what does that look like? Here you can see the deck of a particular liquid handler, and ANTHA then works out from the high level design of your experiment, each and every movement that needs to be carried out, all of the low level details to execute that. And at the top right, you can't see it here, but there'll be a button once you're happy with that to press execute. It then gets sent to the lab. In terms of results, the models generated against the transduction and transfection data for both DOE iterations agreed well, identifying some key factors and factor interactions contributing to the efficiency of the process. So this is figures A and D. In particular, the models were able to highlight some intuitive factor interactions, figures B and E, and some novel factor interactions, C and F, that wouldn't have been able to be identified without this kind of framework and this kind of in-depth experimentation. And not only does it find positive factor interactions, but also you're able to identify areas of design space that you need to avoid too. What was the metrics behind this experiment? So the ANTHA-driven optimization gave a 3 to 10-fold increase in viral titer. And you can see in the middle image here, this is a logarithmic scale, the titer from the screening stage, followed by the second DOE iteration, refine and optimization. And what was the time savings that were enabled by ANTHA? So manually, Oxford Biomedica would have attempted 40 run DOE, and a lot of that is hands-on. They're experienced users with automation, and however, there's still a lot of upfront work done to write the script, often with a few specialized people as well. So that would have been just over 40 hours just to basically code that script in. However, ANTHA does that all for you. So that takes a matter of seconds, and you can start to routinely churn out really in-depth experimentation very quickly. Not only does this help address the market demand for gene therapies, it should reduce R&D timelines and overall time to market. So this is my final slide. Just to give an overall view of how ANTHA can work in the cell and gene therapy space as well. This is an analytical experiment, a qPCR, and you can define a high-level protocol here with this flexible workflow editor, and you only need to do that once. You can then open up certain parameters for for modification to run very routinely. So here you'd upload your samples, the number of samples, the dilution factors, and of course the standards. You'd then select here remotely the device that you want to run your experiment against. So the Hamilton, in this case, robotic system, as well as the analytical device of the Quant Studio, as well as the plasticware you can select. You press run. This is ANTHA working out a digital representation of your experiment. It gives you an overview of exactly what you need and where. So the labware that you've chosen, how many tip boxes, the reagents, as well as the devices, and you can export raw files, etc., etc. This is the preview, so what we saw earlier. So the deck of the liquid handler and each and every liquid handling step that's going to take place. The important thing here is that because Anther has planned your experiment for you, it has a natural traceability from raw materials to the final reaction plate. And with that traceability, we know what's a standard curve, what samples, etc., etc. So we can now take the data and the metadata from the analytical device or the wider process and auto structure that data back to produce a rapid process insight, basically. And with that, I'll hand back over to Sarah. And thanks a lot for your time and for listening.
Great. Thanks very much, Moise. And just to pick it up there, just before we get going, thank you very much to everybody for a fantastic range of presentations. And know everyone's busy, so to spend the time and effort, really appreciate it. So thank you. So at this point, we'd normally say, can you come down to the front and join the panel? But I guess in Zoom technology, we ask all our speakers to hopefully answer a few questions. We've only got 5 to 10 minutes, but let's see if we can get them through. We had a couple on the chat. So from Anthony, 2 questions. First one to Alison, if I may. Alison, I'll praise you the question, Anthony. So basically saying you've got an amazing organisation there, Alison. But the question is, would the core services have been better maintained and maybe given you more options if there'd have been further automation? And a follow-up to that is, do you see more use of those automation technologies moving forward?
I think the only thing that we could have benefited from is having an automated liquid nitrogen setup. I certainly was quite envious when I went to the Sanger Centre and saw how well theirs was organised. And I think it's just the way that universities and groups are funded. If you've got an institute, it's very easy to centralise and cost those services. Doing it in an institution where you've got 108 academics all getting 2 or 3 different grants and getting funding from different bodies, getting that all aligned is much more problematical. So I think if we could have had some automated liquid nitrogen top-ups, we wouldn't have had to consolidate that service. But certainly academics were quite resistant at giving up control of what they see as their samples. But they've been very happy with the service that we gave in terms of consolidating all of the dewars. We had 3 different locations, so 1 in each building for that. All of the other services we provided, I think the human touch was actually required. We personalised our service. Our techs were going around and talking to all of the people in the labs, and it was very reassuring for them to have that presence, that human presence, and to be able to talk through their experiment and what they needed and when they needed it and could we get it ready for them. So getting people to collect things and put them in autoclaves and return it back to people in time, I think that would be slightly impossible to make it as robotic. I think the human touch was needed, but certainly for the liquid nitrogen, I would have preferred if we could have a centralised facility and systems for that.
Yep, sounds good. Thank you. And the second part of Anthony's question is towards Mike. Mike, I think first and foremost, I think the Anther presentation that Moise gave has brilliantly illustrated your original example of the Mini to the Formula One car, because I suspect any attempt to get under the hood of the Anther system would be very difficult. But the specific question, Mike, if you're there, is you talked about training. How do you think it's possible to proceed, though, really, given the important function that academia has of teaching first principles knowledge? I don't know if Mike's still there, is he?
Mike, you need to unmute yourself.
Okay, sorry. I think it's a really good question. At the moment, the kind of bedrock of research training happens through a PhD. And that's, we regard it as a research training period for our students. And increasingly, they're moving away from the kind of research training that was traditional. If I give you an example, people in my lab who are doing PhDs with me, I'll expect them to know what the pH of a DNA extraction kit is. I'll expect them to know some of the reagents that they're dealing with, because those are the questions that are quite often come up in PhD Viva exams. Do you know what the kit was that you were using, and so And the more we go to a kind of more automated, digitized and core facility based kind of research, the less easy it is to be able to do that. And I just feel it's a trade-off. And you know, the example I gave when I was speaking, that actually the skill now in research is all around how to handle the data, not necessarily how it's being produced. I think that's one potential way of looking at it, but I think it is a concern. And we certainly want to turn off that pipeline of well-trained students coming out of universities who really understand the nuts and bolts of the research process and what they're doing in a laboratory setting.
Yeah, actually, it's interesting as well, Mike. I guess also part of the education process is what important. I mean, I certainly remember spending hours hacking HPLC columns and being extremely frustrated that I really wanted to see the results rather than the hacking activities. I learned how to pack an HPLC column, but I'm not sure how much it's really stood me in good stead.
There could be a job for you there, Steve, if you ever wanted it, maybe.
Sorry, did you say that? I missed that, Mike.
There could be a job for you there somewhere. If you...
I'll bear it in mind. Thank you. Nicoletta, I think there's one there for John Marie. Do you want to?
Yes, sorry. Jamie, have you faced any resistance or any support with regards to your application?
I think most people think the idea is very sound, and it's very difficult to not admit that. But now in practice, it is difficult because people are very protective of they obviously got a grant and they got their money, and they made a transgenic mouse, or they have a mouse or whatever organism, by the way. And then they kind of like, they want to make sure they are the one who benefits from it. So there's a change of perception, and maybe it's like, so we thought, you know, the younger generation, the students are the one who would drive this change, but the people who get the grants are more advanced in their career, and they maybe have a more conservative approach to it. So it's genuinely complicated, and that's why this app needs to be quite slick and not take time and be a bit fun, I think. But the idea would be, to share the resource and actually get, once a mouse or any kind of animal model is created, that it publish, you know, there's lots of papers that can be published out of it.
Yeah. Thank you very much. Moise, the same question to you.
Yeah.
Do you have any support, any or any resistance to your technology?
Yeah, and I think actually, Nick, I'd just add to that. Do you see any? It just seems this fantastic technology. Do you see any roadblocks to introducing it widely?
Yeah, I mean, we deal with kind of labs at all stages, really, and I think while we're transitioning, I mean, dealing with some of the large research and development labs, we're often, from the fact that they're labs, we don't connect them to the outside world, and there's a hesitation, of course, to make that available, kind of open in terms of hacking, security, data protection. So I think while we're actively having to work through different kinds of IT setups, ways to connect through different firewall settings and IT security networks to be able to really use this kind of cool technology to be able to automatically, like you said, to automatically structure data so we can generate some meaningful insights, or what it means, not just collect data for data's sake. So I think one of the barriers I would say is being able to connect our lab securely, to be able to and remotely connect. I think I'll stop there.
Thank you, Moise. I think I'm conscious we're out of time, but we've got one more question in the chat Can we just ask that, just time to ask this one more question before we go to close? So it's for Mike from Salman Rahman. Do you think the current changes to work patterns will lead to the maintenance, or more importantly, enhancement of R&D productivity within life sciences and more general productivity across the entire economy? Sorry, Mike's still there.
Yeah, I think it's a very good question. I think it has the potential to, is what I'd say. I think, you know, the benefits of remote working, for example, are self-evident when it comes to reducing commute times for those who've got responsibilities at home and so on. And you could envisage that that could lead to a greater level of productivity. I think there's a big caveat to that, in that what happened, certainly in the academic sector in March, was that everybody suddenly flipped to working remotely, working online with laptops occasionally balanced on their knee on the edge of their bed. And that is not a sustainable way to do it. I think if we were truly going to move to a flexible way of working, then there's two things you need to do. First of all, you need to properly resource it so that people can work effectively. And secondly, I think there needs to be an attitude shift, because hitherto, there has been a mistrust, I think, about people working flexibly and working from home, thinking that that's not going to be very productive. And I think what we've seen is actually it can be very, very productive. So we have to get over those two things. I think there's a level of trusting the workforce that they are able to do it, and there's and proper resourcing of it. And if we can get those two things right, then I think there is potential for much greater levels of efficiency. Great.
Thank you, Matt. I think on that basis, we're a few minutes over, so I think we better try to draw this to a close. Just like to say thank you to everyone for today. Nicoletta, have you got any closing remarks? Or
thank you, Steve. No, I was thinking overall, what are the emerging technologies for the future? It's something that we all need to think how these technologies can benefit and help us, can help the whole research benefit in the future in the next 20 years, and the environment, with a high consideration for the environment. This is something for all of us to think. And yeah, I didn't want to keep people any longer from the dinner or fireworks display.
So Sarah, with that, are there any housekeeping or last minute things? I think there was a survey. Anything else?
I just wanted to thank everyone very, very much for joining us. Thank all the speakers for giving their time, and the attendees for joining us, and confirm that we will send a recording out with a post-event survey and so forth. And to Nicoletta and Steve, thank you very, very much for moderating.